Thank you so much. Come to the second day of the concept workshop. And yesterday we saw how simple chemical systems, starting from chloroperoxidase to cytochrome P450, established the context of Murburn concept in vitro and then in vivo systems. And then I went on to the bioenergetic systems of respiration in mitochondria, and then photosynthesis in chloroplast. So now the context is now set for exploring more complex human-centered physiologies. Now, till now it was organelle, simple cell, endoplasmic reticulum. Now it's more complex physiologies. From simple chemical reactions, metabolic reactions, we are moving on to complex biophysical, physiological outcomes. So today, in the third session, first talk, I'll be dealing with the visual transduction process and neuronal relay thereafter. Here, I'll be dealing with basically two papers, primarily one paper, Journal of Cellular Physiology paper that was published in 2022. So, if you see, the photosynthesis process invoked the involvement of electron ejection out and then movement using oxygen-centered radicals or even other one electron species that are freely available in the medium. Now here in the you see photoreception also has in the chloroplast you have carotenes, right? Like chlorophylls, there are carotenes in the leaf chloroplast. Same carotenes are present as retinol, the retinol, cis retinol, trans retinol that are seen in the eye. So the structural, the visual transduction element in photosynthesis is chlorophyll and carotenoids. Same is the case in visual transduction process. So there is in the visual spectrum pi electron system interactions in photosynthesis. Same is the case here. And another common thing is, if you are aware of physiotherapy, there's something called photodynamic therapy. In that, it's been noted that if you irradiate your skin with far red light and red light, and you get ATP synthesis, right? So just like the photosynthetic system in plants, but it's in a human body, right? So with that, let's start the content of today's talk. Yeah, basically, you know the structure of human eye, right? Eye has the lens here and the retina here, right? This is the retina. Now the light passes through. This is more or less transparent. All these structures are transparent, and then a real, a virtual or inverted image falls on the retina. Basically, here it falls. Now, the retina is approximately what you see here. This is the visual place where the signal is taken. Here it is approximately 400 microns thick. All right. So 400 microns thick, and so these are the kind of cells that are present. Multiple layers of cells, amacrine cells, horizontal cells, and at the distal most part of the retina is the rods and cones, right? They are supposed to be the structural functional unit of vision. They transduce light, and we have the signal from there. So the different the architecture of the eye is there's high amount of vascularization on this side of the retina as well as this side. This side is the retinal pigment epithelium, which we call RPE, and this are the rods and cones, and these are the discs. They highly flattened, just like grana in chloroplast. You have just like that in the rod and cones also, just exactly like chloroplast. In yesterday is a slide I showed you the structure of flattened discs tap on each other. Same here. So now rods and cones are deemed to be the fundamental units of vision in a classical paradigm that you see now in textbooks. And so this has been also given a Nobel, and this is the way the signal, it's called the visual cycle, so as to speak of. What happens is, now rhodopsin is a protein that has this cis retinol, 11 cis retinol. This is the structure. This is a carotenoid, retinoid. Okay, so now what happens is, this thing binds in a particular conformation in retina and rhodopsin, and then there's a cis base formation, and then you have the photon. When the light hits, the photon hits, you have basically a configuration change of 11 cis to all trans. This is the fundamental process that happens in the classical pathway. All right. Now, what happens after this is you have the retinol getting ejected, and that leads to some change in conformation. This rhodopsin, I will show you the structure in the forthcoming slide. Rhodopsin is associated with a G protein coupled receptor, GPCR. If you know, GPCRs are one of the most important pharmacological targets because they are membrane-based receptors. There are a wide variety of drugs designed to target GPCRs. So rhodopsin is found along with this GPCR called transducin. As the name suggests, it's supposed to transduce the visual signal into some other signal that our body can perceive. So basically, now what we have is, in the classical signal, what happens, relay, what happens is, once this happens, the transducin is generally bound to GDP and it exchanges it for GTP. This is very important. For some reason, once the light forms, there seems to be some transducin senses something has happened to rhodopsin, and then it exchanges GDP for GTP. That's the classical pathway. And then what happens is, GTP is exchanged, leads to activation and conformation change of transducin, and then transducin binds to phosphodiesterase, okay, a particular type of phosphodiesterase, thereby initiating a cyclic GMP cascade. That's the classical pathway. Now, here, what happens is, once the retinol goes out of rhodopsin, it's supposed to, now this is the rod cell, right? Now, it's ensconced by the epithelial cell. I'll just go back. Here you see, this is how it is. Now, this is the retinal epithelium. So now you see, in this classical pathway, this has to go into this cell, all right? They're all trans, and then it has to get converted to 11 cis retinol, and then come back to this. So this yellow thing that I marked happens here, and this whatever color this is here. So this process takes hours. This cycle takes hours. It's very important. Now, another important aspect, and this is the structure of the rod cell. And now you have seen how it is articulated with the rest of the composition. So now you have rod cell. The light comes through like that and hits here, and then the action potential, that is the signal from the rods, they go like this, and then the nerves go out of the eye. If you see the structure, that's how it is, right? See, the nerves have to go out through this basic opening. All right. So this is the architecture of the eye. So yeah, you got the fundamental classical pathway, the structure of the rod cell, the cycle, and the overall signal transduction cascade. Okay. Now this is the difference. This is the resting state, how the rod cell looks like in resting state, and when the light hits, what happens is transducin binds to GTP, which binds recruits cyclic, what happens is ion channel is open, calcium and cyclic GMP is there. Then what happens is here it gets converted to GMP and this is activated. So multi-molecular, I told you, transducin has to sense something with rhodopsin, and then rhodopsin, after that transducin has to detach and some deterministically go and bind to phosphodiesterase and thereafter activate it. So in the molecular mechanistic schemes, this aspect is somehow, uh, I would say it's very deterministic scheme, multimolecular, when there is no reason given. So here is the rationale once again, rhodopsin. So light form hits, and then you have trans-retinol coming out. The GPCR gets activated. GPCR is the transducin, which is bound to GDP. It gets activated. It exchanges GDP to GTP, and then GTP is active, and this binds to phosphodiesterase, and then cyclic GM giving, and then this activates cyclic, utilization of cyclic GMP by the phosphodiesterase. That's the classical scheme. So rationale for exchange of GDP to GTP is the key step in signal transduction of GPCR. All right. So, and there is no rationale given, and this is what it looks like. So, can I take the questions in the end? Because we have scheduled to I'll answer all your questions, any extent. So, subsequently, we are dealing with the neuronal propagation, right? The impulse propagation along the nerve. So, the classical scheme for which again a Nobel was given was the Hodgkin-Huxley model, if you know. So, here is the basic principle of it. I'm not going to go into the detail. So basically what happens is there is a resting potential, and if the signal is low, you have a graded potential. It doesn't completely fire the neuron completely. It doesn't go through completely. You have a small signal, but when the signal is of sufficient threshold, what happens is a series of events occur in a very deterministic manner. All right. So deterministic manner is the sodium channels this neuron. Now this neuron is these axons you see here. These are called axons. All right. So they are supposed to propagate a signal, and that signal is via this modality. That is sodium on the surface of the neuronal membrane, axonal membrane. You have sodium potassium channels, and then deterministically sodium channel opens, potassium channel closes, you get depolarization. Then sodium closed, K open, you have repolarization, things like that. And you have at a given point when you measure the transmembrane potential, you have a variation of the potential in this manner. All right, you get that? That is what the classical theory is. Now, here I want to say here, this is the Goldman-Hodgkin Katz equation on which these things were developed. This also was recognized. Katz also got a Nobel for this. So basically, the sodium in and out and chloride in and out, all these give treatment of these things alone are deemed to explain the transmembrane potential, fluxes, variations, and all that. Ionic fluxes, variation. And basically, what you do is you're considering the cell as a simple solute system inside and outside, and then you get a potential variation purely due to some ions being pumped from inside to outside and outside to inside. All right. So now this has been mathematically modeled in the classical model like this, but the same form of the equation, my collaborator, Hirohisa Tamagawa from Gifu University, Japan, he's got the same form of the equation, you see, but then he started, rather than with ion pumping mechanism, he started with the theorization of an ion adsorption mechanism. Who was it yesterday? Adsorption isotones. Yeah, he should have been here. I said that I'm going to talk about it. So So signal relay here is by an ionic plus transmembrane potential based ripple along the axis. A ripple, a kind of a wave front kind of propagates owing to some uh, ion fluxes, sort of. Can I take a little bit of your water? Thank you so much. Yeah, thank you. Yeah. So now, next, as usual, the questioning part, right? This is my favorite part. I always like to, when something don't appeal to me, I like to question. Yeah. Now you see the architecture of the atom. It's something that strikes very significantly. Now, this is the structure of retina, right? There are at least 10 odd layers of different, I just put it in different colors just to show that there are multiple layers of different optical properties, right? And very many layers. That's what I've done here. That's what it is. So here you see the action potential originates here, and then it has to propagate like this, and it has to come and go here. Now, if the classical theory were to be true, and evolution is an operating principle, what would you imagine? You would imagine that the theory states that rods and cones have evolved to tap diffused light. Because now here is the rod and cone, right? They are oriented towards this side. Their orientation is here. And now they say this kind of shy. It doesn't want too much light. It's taking only diffuse light that has already come through multiple layers. That's what it is. Now the point is, if I were to imagine that an evolutionary mechanism is involved in forming an eye, I would just, if there is a diffuse light that gives a better transduction, I would just put a diffuser here, as kind of a membrane, if you may. And then I would have the rods and cones facing here, and the retinal pigment epithelium like that, and the nerves to conduct like this. Did you understand what I'm saying? It's a rather simple way. Why have such complicated things which will mess? Basically, if you imagine 400 nanometer is the starting point of a visual spectrum. 400 nanometer, if it has to traverse 400 microns, it has to go through 1,000 times its wavelength, right? So by the time it reaches here, what will happen? Scattering, diffraction, in all kinds of wave phenomena that you can imagine, whatever I've written here, absorption, reflection, and all that. So suppose you imagine an eye can meet a photon coming from Amit's side at a particular point in retina. Okay. Next time the same, it will not come from there. It will not have the same optical property. An evolution mechanism cannot ever reason with transducing such a signal mechanism, such a mechanism. Wherein the bottom part is rods and cones are the fundamental visual transduction unit. How can it be? Because by the time, whatever signal that is coming in, that signal will be completely scrambled, basically. I don't know if I'm using the right word, but that is what I came to my mind, basically. So now I come to the structures of the proteins that are in. All right. Now, first we looked at the microscopic structure. Now I'm looking at the protein structures per se. Here is the rhodopsin. Here is the transducin. You see, this is how it looks like. All right. The rhodopsin is a simple Damru type of structure. She was that thing, no? It is something like that. And it has the retinal here, like that. And this part is located in the lipid membrane. Okay. And now here is alpha, beta, and gamma structures. There is once again like complex 3 of mitochondria. The beta has no attributable role. They don't figure out what it is. Please pardon me when I use they, because it's a very simple way of identifying the classical they and the alternate mechanism I or Murburn. So so here you see the aperture, the beta opening, all that here, these structures. So here is it in elaborate region, this thing. Now, classical methodology says that alpha and the GDP and GTP bound protein have some conformation change, right? That's what the transduction methodology is. And then this, once it's bound to GTP, this alpha-transducin, what it does is it can go and bind to phosphodiesterase, this puppy here. Now, the problem is this transducin bound to GDP and bound to GTP has been characterized. Practically identical structures. Practically identical structures. You see the superposed, there's hardly any difference, whether it is bound to GDP or GTP, the alpha. And like I said, what is the beta? This is not known, right? Sitting here, that hole is seen here. That now, that's one. Now, how can something whose structure is known, which shows no difference, somehow give some signal downstream, activate it like we don't know. So now I'm going to just ask some questions, right? Now I has been innervated on both sides. Why? There is no role for oxygen in the visual process when you know very clearly when you have low oxygen pressure, you have blackouts. You cannot see. Then proximal and why do we have vasculation on both sides? Higher lights, why you have more light impinging on retina, you get more dross. You remember the terminology from last, it is diffusible reactive oxygen species. Why more light gives more dross? Then why do we have photo bleaching at high intensity? Light too much, your eyes get bleached, right? Basically, it's just like any other process. So it loses sensitivity and ability to function. Then recycle time and relay efficiency. The point is, RPE recycle is slow and wasteful. You have rhodopsin coming out, it has to go. It is slow, it is wasteful. One photon hits, after that rhodopsin transducing thing is gone. It is taken out of the picture, right? And then it has to go through a whole huge cycle, which comes back. Then, so how can the retinal visual cycle afford a dynamic kinetic resolution of 1/60th of a second with subsequent photoresponse recovery? That is, we see dynamically within 1/60th, we can process images like it so quickly. Whereas we know that each it is the subsequent photoresponse. That is, the process is slow to, for the classical scheme, it is slow to respond. But in the real life, we can really process image very quickly. Then one important aspect about relaying is, the neurons, they have something called myelin sheath, and the myelin sheath, when the myelin sheath is present, signal goes faster. Why? Because you want the ripple, if the ripples are, without the myelin sheath, it will go faster. Why is the ripple, which is covered by the Schwann cells and the neuron, when it gets covered, there is no real explanation in the classical school why covering the song with the myelin sheath. That's good. Why does it give faster delay? Thank you so much. Then, how does this signal get converted from a photo to an electrical signal? Right? That's the next part. So, transmembrane potential variation affected by isomerization, ejection of retinal, and conformation change of transducin. How is that happening? How is that conformation change giving retinal ejection, making a conformation change in transducin? And we have seen that transducin, there is no real change. The transducin change, how is the transducin ability to bind GTP and GDP changed? How? Why does rhodopsin get phosphorylated or oligomerized? There was one Dr. Prakrit who wanted to know about the phosphor.
He's online now.
He's online. Okay. So now that part, again, multiple phosphorylation of rhodopsin is seen, right? So Then resolution, and this is very important. Professor Amit would be interested in this. Rod cell packing is approximately 2 into 10 power 5 per millimeter square. You know, the pixelation kind of an analysis, that's what is being done here. And 30 to 60 arc sec is the cell dimension. And if you take the curvature and try to study the angle subtended on that arc, that's what this arc sec is. So the eye's resolution is approximately 5 arcs. If rods and cones are the fundamental units, they themselves are of the dimension of approximately 10 times the resolution. Is that possible? You ask yourself, is that possible? How can I have a resolution of 5 arcsec when my measuring unit, the detection unit, is 50 arcsec, 10 times that size? Arcsec is, suppose now you have a small football like this, some distance like that, you subtend and you Google it and see.
So that's subdivision of a degree.
Some degree. Okay. Now, okay, I can give you an exact terms. Can we come to it later? I'll show you exactly. Yeah.
It's a solid angle.
It's a solid angle.
It's a degree and then minutes and then seconds.
Yeah, degree. That's just solid. That's the. How can a single row of rod cells located at the distal end of retina afford depth perception? Yeah, you have a depth perception, right? The logic of depth. I know Chinmay is sitting here, Professor Amit is sitting here. How do I know that there is a depth aspect involved? Even for the brain, even if it is at a analysis part, how does that come in? So, what is Murburn model of vision and signal relay? Basically, so like photosynthesis, you have basically the activation of retinol giving excited retinol, and then you have superoxide formation in presence of oxygen. There is system crossover and all that. I'm not going to the photochemistry in detail. I'm already lagging behind in time. So now here is the recovery of the recovery reaction. Here is the activation reaction. You can see once the thing happens, this can, once again, the ROS-based reactions can easily regenerate oxygen and, not completely, but the process is reversible with ROS also. So photoreaction involves the heat transfer from retinol. ROS formation occurs in the vicinity of rhodopsin, and lost electron can be recovered subsequently from milieu via multiple modalities. So here it is. Now here is the theory. Active rhodopsin, oxygen, F gives superoxide. You have a transmembrane potential variation. Here is a charged species production, mobile charged species. Now this superoxide can attack GTP and phosphate to make GTP. So this GTP active transducin can modify it and release GDP out, and then this GDP can come and bind to the phosphodiesterase, leading to the activation. So that is what is written here. Effective charge separation and formation of superoxide leads to hyperpolarization. The production of ROS results in phosphorylation of GDP bound to transducin, and thus formed GTP is hydrolyzed and released as GDP by transducin. GDP unclips gamma module on phosphodiesterase 6, activating the cell. Now I'll show you the overall scheme for both concept. Sorry, I don't know what's happening. Yeah. So here, what happens is here, it makes ROS. So this attacks GDP bound transducin here. It makes GTP. This GTP is hydrolyzed, giving this. Once again, it gets back there, and then this GTP gives out GDP, and then this GDP unclips This is the explanation. Now I'll show you. I'm not going same explanation. I've just made a very simple cycling. So basically, oxygen needed, no dissociation of retinol. You see, there's no dissociation of retinol. Retinol can dissociate at high light and all that, but that is just a wasteful or some other reaction that can happen over a period of time. All right, it's not something that is the direct process. Then GDP is the signal for phosphodiesterase activation and not protein-protein binding. So, and alpha-transducin is a GDP phosphorylase murzyme, and beta-transducin is a GTP hydrolase in the new theory. Now I'll show you. And here is the Murburn model of signal relay. Rather than an ion flux, it's a movement of electron along the axis. So if it were an ion flux, how do you explain that there is no radial dissipation, no backward dissipation? We know that axons have a polarity. All right, axons have a polarity, and I'm saying it can go along the membrane surface, or it could even go along, say, a tubulin or something like that. Here I've just showed one of my rationals, and here also you can have simply, you can see here based on oxygen-centered reactions how the transmembrane potential can easily vary from in a temporal manner when the oxygen goes from hydrogen peroxide, oxygen goes to superoxide, hydrogen peroxide, and water. This simple redox cycling can also give transmembrane potential variation. That's what is shown here. So basically, as per this theory, rods and cones are the final electron source. Multiple cell layers could transduce photons. All right. So that can give depth perception. All right. And higher resolution because you have more number of cells perceiving. Okay. Rather than just the retina. And upon stimulus, E movement from dendrites to synapse, and action potential is a sign of E movement along the axis. The action potential flux, that signature that you see, it is a variation of current actually in the system flowing through the system. So when you measure it across the cell, you can see it like that at a point. That's what I'm seeing. Now here you see, here is the simple cross section of a retina, resting state. So now you have a ganglion cell, for example. Here in my theory, all these cells are photosensitive, not just the rod. All these cells. In other systems, in basic animals, simpler animals, these ganglion cells are the photoreceptors. Why in human beings? Why do they only the rod cell? We have no explanation. So I say that this is the case in human beings also. What happens is here the nerves take the signal away, so they become electron deficient. That is served by the rod cell. So rod is like a electron pumping kind of a machine. All right, and then there is this dielectrics, this lipid. They are evolved to somehow this kind of a structure. There is ordered some chaotrope and cosmotrope. They are molecules that need in the ions arrangement that can be kind of perturbed in a systematic manner. There are arrangements and molecules and ions like that. So this is the signal relay wherein electrons are transferred along neurons and brain becomes the ultimate acceptor. So basically, sequential temporal events for ease in compression, I've shown it like that, but it occurs actually parallel. So basically, there are gradients, polarity gradients, and the polarity gradients affect the signal relay. So evidence for the Murburn. This is the simplest thing that supports simple structure of rhodopsin buried with retinal deeply buried. Okay, with protein in the lipid phase, non-conservation, and this protein structure is not really conserved across animals. Okay, there is a significant variation. So if this conformation, this binding conformation change is very important, how would you have this fold not conserved? That's a question. Then oligomerization of rhodopsin enhances, right? In the eye, it is often seen that rhodopsin oligomerizes. So I have said yesterday that, when you work like that, you want the radicals to be produced in close proximity so that they can be effectively utilized. So that's what it is. And now you see the chemistry of the process. I have not yet modeled the physics of the process. All right. I've just shown this kind of reaction is you start from the delta G formation and you see the overall process very viable, very, very viable. And so here is the beta transducin. Here the structure, there is no change. You see the opening here very clearly. GDP bound, see, KD of GDP is picomolar. You see this, very important, picomolar. 80 to 300 into 10 to the power minus 12. Okay, that's the value here. High binding, very high binding. Here, GTP is 0.5 to 0.6 millimolar. So how can it exchange? Whether it is, both conformations have the same kind of values. Did you understand what I just said now? That is, in the classical theory, upon hitting, there's some kind of a difference. But then we took both structures known. We docked with the GDP and GTP. They give for GDP very high binding efficiency. So how can GDP get exchanged? It cannot. So it will bind to GDP. So little difference between GTP and GDP bound proteins. For the GTP has much lower affinity for the protein than GDP. How can something that has a lower binding efficiency get exchanged in lieu of the higher binding efficiency? So that can. So here, what for my theory, what happens? The superoxide that is produced attack the GDP, it becomes GTP, and so naturally it will release. Yeah. And once the GDP comes here, you see the structure beautifully. That hole there, it comes from this side, and it attacks with it's a GTP formed, comes and attacks. It is attacked by this beta-transducin. You see, GTP has a binding of 2.5 micromolar, GDP has 1.5. Beautiful, no? So with simple docking, we are supporting the structure. I predicted this, that it would be happening. And I asked my student to check my model, and there it was. Bang on, bang on. Exactly as per prediction. That's the beauty of the physiological model. So, beta-transducin, now you know the role. Clearly. So, tangible enzymatic functional role of beta-transducin and agrees well with the structure and in silico findings of the story. So the overall structure is beautifully explained with the new model. All right. And now you have the GDP that is released can go, and now very clearly, this is the structure, right? There is a clip like this. I showed a clip, this yellow thing, right? Right, like that. So this is cyclic GFP binding site. So what happens is it has two sites actually. So basically this covers that. This is the site that is covered. So now when GDP is present, GDP binds these places. So it unclips the thing and makes the phosphodiesterase happen. So once again, the perfect agreement with the predictions of the binding concept. So once again, this signal cascade is beautifully explained. Then the transmembrane potential. My collaborator Hiroiza Tamagawa again demonstrated for the first time that transmembrane potential can be changed in vitro systems with radical production using enzymes. So basically, you have a simple setup like this, and you take membrane potential across a transmembrane potential by generating. You have an enzyme and generate a fraction of a second. It comes and you get a change like that. In the control, you don't get, and this thing gets perturbed significantly by flushing with nitrogen. So, and also, Hero established that TMP varies due to membrane charge, and it varies due to the number of membranes, and TMP varies due to the ion in the path reference and all that. So, transmembrane potential is not really a very simple thing, as simple a few solution ions in solution moving here and there. It's a complex phenomenon. All right. So paradigm shift here is that seeing vision, you have to see vision differently. Vision requires in the contemporary textbooks. There is no role attributed whatsoever to oxygen. Without oxygen, you cannot see. All right. Then role of other cells in visual construction. Rods and cones are not the fundamental. But then if they have to continuously function, they need rods and cones. And that's the reason the rods and cones were deemed. So new modality of signal relay. Rather than seeing it as an ionic flux, in the next talk, I'm going to talk about how can ions be pumped deterministically. For 1 second, I think I want to pump sodium. I can say I like laddu, I don't like jalebi. All right. Protein sitting there. For 1 second, I feel I have to pump sodium. Now I have to block this. I have to pump potassium. I have to open this. I have to let potassium in. I have to. How can that logic be justified? They say it is because of potential variation. Then the potential is varying because of the ion. Now you tell me what is what. Did you understand the argument here? No, that is the potential controls the ion, and the ions control the potential. Then who controls these things? What controls this? There is no real chemico-physical logic that is tangible here. So that part here is what I've written here, the paradigm shift. I would like all of you to peruse, if you're a research scholar, peruse the work of Federico Faraci. He just did his master's. And, I found his master's thesis from the a German university, Leiden, I think, Leiden University. And I wrote to his professor and found this person. He's not traced. Then I found him and then we networked. He had critiqued on the 60th anniversary of Hodgkin-Huxley model. It is the standing physiological model, Nobel recognized. He had the guts, and it's a very systematic work. If Federico is hearing this, I congratulate you for your wonderful work. And that's what this kind of research should be. You should not really bother whether it is a Nobel recognized work or not. It should appeal to common sense, and that's what is important. So how does an ion ripple propagate based on some deterministic mandate is something that I could never understand. Even when I was studying my master's, I could never buy it. I could never understand. And I had to bear the frustration of having to teach it also. So I was a biochemistry teacher. I had to teach photosynthesis, the old model, the new model. I had to teach it. But then, my student, I felt the same doubt when I was in countryside Kerala, Nita Unnikrishnan. She felt the same doubt. And she asked, sir, how is this thing? I said, I also felt the same thing. So if people can critique question like that, then it leaves a jarring effect, it's not sitting properly in the brain. So that's how you question. So, once again, and then Daniel is my research scholar. So University Leiden, yeah, here is it. I would strongly recommend, if you're a research scholar, to see how systematically Federico went around, Faraci went around analyzing what a scholastic piece of work this is. I don't speak that scholarly. He has systematically dissected the why sodium movement need not be. Some other movements, in Tasaki, and there are so many Japanese workers in the 60s and 70s that conclusively demonstrated that these kind of theories are not right. But yet, these theories are taught. It's a sad thing for human beings that we still have to do. It's very sad. It's very sad. And I complete my first talk on this sad when people like Federico Faraci are not recognized for their critical, beautiful, systematic work questioning that. And he has based it on solid experimental work by so many other researchers. So if you have the time and the interest, and if you are into this business of physiology of neuron function and all, how can you understand how the nerve functions, how all the diseases you have, if you work with a sloppy false mechanism? You cannot understand. if it had to get recognized, or if the research has to work along these lines, those people who are working along these lines should be willing to change or try this new approach, right? So you would imagine that this is recognized with a Nobel, it is being taught. You would imagine that there are, at least in my review, I've quoted at least 10 different neuronal signal propagation mechanisms, 10 different. So I believe that students should be exposed to all these schools of thought rather than make them parrot information that this is happening like this, that is happening like this. What? So with that, I end my first talk. Thank you so much for your attention. I'm open to questions.
So,
Yeah.
so in one of the slides, showed that along with this ganglionic cells are also important and they have some different state. So, did you have some at least a bioinformatics study and then looking at genome and whether there is a
Wonderful question. Wonderful. Chinmay asked me that my theory proposes rather than rods and cone cells being the fundamental unit, I said it is like a powering unit, and it is the fundamental units are somewhere in ganglion cells and all that. Did you do some work to check out the melanopsins of ganglion cells and all that rather than rod? No, I did not. That is something that anybody, that's a wonderful question, and that's what should be done. And I left it at this, saying that in all other animals, systems cells are the known photosynthesis. So and man with a lot of limitations, with only explorations, at that. So that I'm just trying to say that if you want to, anybody can. That's how this field should go proceed further. Wonderful question. Yes.
So I have a question. So in the classical way of looking at how the retina works, they say that rods they sense the intensity of light and cones sense the color. So in the alternative model,
Oh, wonderful.
how does that work?
Oh, wonderful. I have no idea. Outset, see, I have no clue. I have to read what cones particularly work. I have no clue. Because three cells are supposed to give the um, three types of cone cells. They say there are, right, something like that, right? We are able to differentiate so many. As of now, I'm just sticking simple phototransduction mechanism. It's a wonderful question. Wonderful question. I have no clue, and I'm honest enough to
Dr.
that.
Dr. Murali, can I ask a question? Yeah, Professor Lakshmipati. Sorry,
Yeah,
I
know,
couldn't
yeah.
join you yesterday. One
Yes.
question.
Yes,
Well,
please.
it is a combination of 2-3 questions. So I may take a minute or two to pose that question. Number one, whether it is retina, muscle, or any other thing, it is actually neurovascular epithelial endothelial cell systems coordinately working together, right? Second important question is ATP versus GTP. The classical models always say that ATP can be exchanged with the GTP. I am not convinced of that thing. There are several evidences for that thing. Third important thing is ATP production itself is controlled by RBC. You are talking about what is called in your Murburn system the hemoglobin role actually in this thing. We have two levels of oxygen availability to the cell. Number one is cyanide resistant oxygen. Number two is oxygen that is consumed at the level of complex 4 in the mitochondria. Right? Now, where from this particular cyanide resistant coming? Oxygen availability to the cyanide resistant cells, that is cyanide resistant respiration in cells. That oxygen availability comes from the hemoglobin breakdown, right? Hemoglobin breakdown is controlled at 3 levels. One is 2,3-phosphoglyceric acid within the RBC system will be actually controlling that particular oxygen release from that thing, and that actually contributes to your NOx4 and other systems also, where actually. Second important thing is, what is the source of iron when actually this is coming into the picture? Invariably, whether it is a muscle cell, retinal cell, or any other cell, you have what is called hemoglobin breakdown, that is availability, that makes it available for iron. Iron is important for activating shift from sodium to potassium also. It is also activating shift from anaerobic system to aerobic system. That is where actually again aerobic systems are partitioned between cyanide resistant and cyanide susceptible. Cyanide susceptible systems are those which are present in mitochondria. Classical models, whether it is Klingenberg model or Chance model, they are prisoners of classical models only. Therefore, what they say is ATP is exchanged to GTP. That is not correct. For the past 5-6 years, if you are carefully looking into what is called the mitochondrial structure function, VDAC downregulation activates cardiolipin biosynthesis. But VDAC is also essential where actually lipogenic systems are also activated. They actually activate this thing also. I'll just take one more minute, not beyond that thing. So GTP activation takes place only when the mitochondria assume the globular structure, the so-called double membrane structure. I call it so-called double membrane structure because watery layer around that could be mistaken as a separate layer. Okay, that is one aspect which we need to examine, and then research is coming out in the past 5-10 years. VDAC versus cardiolipin biosynthetic programs, vacuolar systems exporting. That is, how do you actually integrate this particular system of neurovascular systems plus myelination to that of what is called your model of ATP synthesis. In my opinion, ATP synthesis is three-phase system. In somatic cells, normally resting cells, there is no need for much of the ATP. In mRNA synthesis, you require ATP, and DNA synthesis, where P53 is activated after the restriction point in cell cycle, you require actually this ATP formation at a higher level. Thank you.
Thank you, Professor Lakshmi, for the wonderful. Yeah, so I very sorry, I really couldn't catch the extent of your question. I think your question deals with our earlier interactions about my model of ATP synthesis per se. So, and with respect to the vision that I was talking about now, you are asking me a specific question in the sense that how does your model fit all these complex cellular systems with respect to their complex functioning? Well, this is at a very rudimentary stage where we are. I've just challenged the fundamental transduction procedure. I've challenged the mechanism of rods and cones being the fundamental units. I've just challenged the way transducin and rhodopsin are supposed to work. These are some of the things that I've challenged, and I've given sufficient in silico evidence and structural evidence with respect to that. With respect to the exchangeability of ATP versus GTP, I do agree with you. It's not that easy, all that, as they say. But then here, what I've shown in this system is that the GDP is known to be bound to transducin. How can it get converted to GTP? That's what I've explained. And then how does that GTP that is converted, how does it get released to serve as a messenger for phosphodiesterase? That's what I've explained. The classical model requires deterministic mobility of these proteins on membrane systems and then identifying with membranes when there is no rationale for any of these. So that's what I have explained in this talk. As far as your cyanide resistant and cyanide susceptible, I do agree that, if you have a cyanide, my theory requires a double membrane
part wherein the proton gets the incoming protons easily get scuttled. The transmembrane ion differentiation also gets screwed because of the cyanide's role. So in free solution, like what hemoglobin is present, it need not go through that. In fact, in the in vitro model, in vitro ATP synthesis, you need much higher cyanide to inhibit that. That is shown. So I agree with most of what you say, and some part of it I could not follow. Perhaps we can have this conversation and take it up at a later time.
Yeah, at 1:30 you said that you have some interacting system. At that time, I would like to talk about this thing, but let us, for the benefit of your students and yourself itself, try to look into three issues that are coming up of late for the past 5 years. Number one, Hemchandra Reddy, who works on actually Alzheimer's all through his life, he was working on that thing in the past. Texas University, Hemchandra Reddy, PH Reddy, you can, Texas University, who works on Alzheimer's.
Yes, please go ahead.
Yeah. He had been reporting in series of papers that VDAC downregulation activates cardiolipin biosynthesis.
Okay.
Number two,
Let's go ahead.
the cardiolipin biosynthesis is also associated with actually lipid biosynthesis and particularly unsaturated fatty acid lipid biosynthesis, which are tagged on to this thing. A recent paper from Hyderabad University and also Hemchandra Reddy's papers will also give you a lot of these things. This particular partitioning between actually the cardiolipin and also VDAC dependent mitochondrial structure has to be investigated more thoroughly. Even Hemchandra Reddy is also not totally out of box. He is still considering that as this thing, but he only says that down regulation of VDAC is this thing. In my opinion, cardiolipin associated with CTP synthesis is the starting point for fusion proteins activation, and that leads to actually the globular structure where GTP is synthesized, ATP is not synthesized in electron transport chain at all. ATP production is three-layered production. Number one, at a base level, it has actually ATP synthesis at glycerol, 3-phosphoglyceric acid. I think one of your paper also says that thing. The second important thing is when actually RNA synthesis is taking place, the second phase of ATP comes into the picture. When P53 comes into the picture, the third phase of ATP, where nucleic acid, DNA synthesis, and then cell cycle propagation will be there. So these three things have to be integrated, and without actually tissue crosstalk between the different cells, we will not be in a position to actually address the biology properly. Ultimately, our effort is to attend to the therapies where actually to cure diseases. Thank you. Sorry, you can go ahead, please.
Thank you. Professor Wadlakonda has got several questions for how we consider the normal metabolic cycles in, that are taught in textbooks. Some established names become